And then second, I want to thank everyone here for showing up. Hopefully you'll attend this afternoon's portion of this session. I hope to see you there. With that said, I'd like to open up the floor now to questions you may have to any of the speakers or any general topics for that matter. I just ask that you use the microphone when you ask your question. So, okay.
Thank you very, very much. First, thank you all for this very interesting session. And I have a question which, well, another question that I would like to throw in the debate, so for all the speakers, which is we all agree, I think, in this room that size should be considered a critical quality attribute. But, and up to now, very often, and also, as I think it's described in the last guidance from the FDA as well, it is accepted to report the average size, for example, by DLS, so just putting the average size and the polydispersivity. You have shown very clearly that to check for batch-to-batch variability, even in cases where you don't suspect it's needed, it's really maybe needed to go further, so to add, so to really consider the particle size distribution, which is acquired by an high resolution technique, no matter which this is. So is really reporting the average and eventually a low resolution parameter which more or less indicates the polydispersivity enough for a critical quality attribute, or should we somehow start, I don't know how, this is complex, but to compare particle size distribution in a more in a statistical way, or at least always report the D50, I don't know. So this is something else that I would like to ask to all of you.
I can, I'll let them comment first.
I think the simplification where you have a Z average and a PI, we're going to hear that now in a talk, it's time to leave and go somewhere else, because it just shows to me the person does not know what they're doing. So it's just a total oversimplification. And the correct way to show size distribution is a 2D chart of number versus size. And then you get the number, and it's not one number for concentration, because that doesn't work either. And you should just put up a proper 2D histogram. And it's quite simple to achieve. It's not very difficult. So my question for Jeff before is why do the leadership in the particle technology world allow that oversimplification just to perpetuate? I mean, it's just time to say no and move on, people, and do it properly. It's not like it's difficult. It's really quite simple.
I don't really have a good answer, but I can perhaps submit that from a regulatory agency's point of view, it is a question of the risk that, or what kind of product are you dealing with, and what's the known, implied, and estimated risk of that particular kind of product. And that really determines what we are asking. And if I may put this in context also with a terminology that we actually don't use at FDA in the non-biologic complex drug world, similar challenges there in terms of characterization. And it's not that we say, choose the most simple method and it's okay with us. And you know, the challenge for generic products is also a risk-based assessment, even towards, and that's kind of a philosophical answer, not to give you technological details about it. For a generic application, where typically we do not request extensive clinical trial studies, it doesn't mean that we could not ask for it if the drug poses a significant risk, or if the therapeutic window is very narrow, or if there is toxicity related to variants in the product. So those options we always have, and it's always considered in the context of the particular product. And I hope that's an answer that helps you. And of course, the discussions with the experts from the regulatory agencies with all the other international groups is an ongoing process, and I don't think that we have reached any of the final answers, as much as regulation is always in flow and we deal with learning systems. We just have to make sure we communicate well and we get on the same page and we use the evidence of science to make good decisions.
And I'll just comment on that, the whole sizing as a CQA. So obviously there's many other parameters beyond sizing, right? So the ideal really, though, is to define your CQA. You need to link that to a biological outcome. So if you have a sample, let's say you have 3 batches that have sizes 10, 12, and 15. Now, does that really mean they're different in terms of biological situation? It may not be, right? In which case, then, who cares what sizing you do, right? It doesn't matter what technique, right? In that case, you would look to something else as a CQA. Maybe that's drug release, drug kinetics, for example. Now, I didn't show that, obviously, because I try to focus only on the sizing aspect, but we look at many different parameters beyond size, and there are many, as shown by the presentations today, to look at. But just trying to answer your question, it goes beyond that, and you really have to link that to some biological outcome when it comes down to it. It may be that sizing may not be the CQA in that case, right? In which then you move to something else. And that kind of leads to my question then, so if that's the case, you have all these techniques coming out there, right, where you can say, I have 5, 6, 7 nanometer, 1 nanometer resolution. How important is that, though, in a biological context, right? Now, depending what your background is, you can debate that heavily, right? Depending on what you're doing. So, and I'll leave that to the group as well to discuss that aspect. How important is that resolution and sizing techniques as it relates to a biological context then?
I want also to add into what Jeff has said and reiterate what also he had inputted into his slide. The use of reference material at every stage on the cascade is also critical to determine, and the comparison with equivalent or with molecules that can actually fit the purpose for the specific measurement also enable to go towards the biological application. And that is an aspect that also FDA has addressed as the European medicine agencies in the way that when you develop nanomedicine, you need to prove to have efficacy. And that doesn't come to only the API efficacy, it's also the carrier has to be proven that is fit for purpose. So the sizing is functional of a proper measurement, as I highlighted in my, and not pressing a button, but also the aspect that you're measuring what you're trying to use in this specific nanoformulation. Because if you are just feeding a matrix of information, you're not actually delivering a good service.
Any questions that you have? I can also, as my presentation showed, I brought up those 3 topic areas. We can always discuss that if you like. Yes, no? Okay. So the one question we brought up was, making measurements in, let's say, a biological relevant media. How often do people here do that, for example? Like, I guess you can raise your hands. Everyone has different backgrounds. Who actually does it in biological media, and how do they do that? I see Hans' hand up there, so and Luigi. So the question then really is, well, let me ask those 2 who raised their hand, what techniques do you do to make, we heard from Luigi already, but how do you do that? And what are the limitations for that method?
We use TWS, obviously, but with heavily biological systems like plasma, we now have a column method to measure exosomes, because we separate out the exosomes from background proteins, because otherwise the media is just not bounded by the proteins. People want the exosome information. So in fact, I'm speaking this afternoon, and one of the talks is about precision isolation of the nanobiologicals, because if you don't precisely isolate them, you can't precisely measure. So it's just another part of the same thing. One comment I might make about size, there's a good question is what actually is the size? What does it even mean? Because you've seen how many different types of particles there are. So you've got a gold particle, you can measure the physical size of the gold. You can use the hydrodynamic diameter, which is a different thing. If you've got ligands, then what does the size mean then? You know, you've got all these little fronds hanging off the particle. But what do you mean by size? Is it mass? Is it the outer diameter of the ligand? Or is it the volume of everything? And the different measurement techniques are going to give you different answers to that. You know, so we didn't discuss that today. This is just one size fits all, if you like.
Yeah, I agree. I mean, that goes back to the whole orthogonal methodology. And I mean, I agree with sizing. There's so many different ways out there. It's like when you report a number, you need to report how. You know, first of all, what measurement you use. And even just take DLS. Everyone here, I think, is familiar with DLS. With DLS, you have intensity, number, volume weighted, you have Z-average. So going beyond just giving a number, you need to specify what size that is, along with all the conditions as well that goes along with it. You know, what temperature, what solvent, concentration, things like that. And that goes to your point, Hans, where, you know, which size is correct, we don't know. I mean, most people will record size with what they have in their lab, right? You're not going to do something you don't have, I mean, to be honest, right? Again, I just go back to that, I hark on that whole thing. It needs to relate back to some biological outcome. You know, reporting a size and then saying, yeah, it looks fine, but it has no meaning in a biological context. It doesn't mean anything. So you want to always link those two together, and I think that's very important when reporting size.
Let me also give you some background, and Jeff can correct me if I'm wrong. More than a year ago, the Nanomedicine Alliance and several of us in Europe are starting a discussion around sizing. And this is involved, as Jeff briefly outlined on the ACS, the engagement with industry to define what are the critical quality attributes for sizing and concentration, because this has become the entry point for measuring quality, for quality assurance as well in their continuous batch production. And we went through the entire spectrum of characterization. We didn't mention about tailored diffusion, for instance, today, which is another technique, size exclusion chromatography, AUC. So we haven't touched base on all that because, as you rightly say, there has to be a window of sizing, and this is what I capture in the table, but also there has to be a tool that could be easily, readily available. Not everybody has a tailored diffusion. If you're a physicist, you can build it with capillaries and 2 micropumps. But if you're a biologist, you need to acquire the system, and it's not come cheap. Malvern has launched a new system called Archimedes. We worked on buoyancies. So if you actually measure liposomes and the carry and the drug loading, that could be another system. So there is a panel and there's a spectrum of all this. And within the Nanomedicine Alliance, we actually dealt with it, and we invited people to give presentation to create a landscape. We hope this is going to be released in a form that is going to be open access to everybody, because the consultation was open to everybody. So, and this is why the 2 workshops are the culmination of all our effort, where we put together question, but also we open the panel for discussion. And questions like this one are very important. What is sizing? What are we measuring? What is relevant for industry? We have Polymon that has discussed it. My question to him was, do you do inline measurement? Could it be this one that improved the service? Or in a batch mode, there's no need to identify the variation in the measurement? Should it be better by an A4 and do batch and looking at the variation in the distribution size versus the concentration? So I think here we all raise our hand and we actually explain the typical problem. But then I go back to the data mining, because the most complicated system produces a large amount of data. So to a measurement, it takes a week of data analysis. And if you have a wrong measurement with wrong settings, you're wasting a lot of time and you don't get the answer that you want. I hope I throw more back into the pool for discussion, because it's very important that we come with an opinion, or at least we come with a set of questions that we can actually then reiterate back and address to the different people that are in the room, and then expand out to the rest of the community.
So, of course, with all that, to use the best science to get the deepest knowledge is certainly a part of it. But when you think pragmatically of making a product, fit for purpose. I would use that phrase, fit for purpose. And I think Jeff mentioned it, for what do you need it for, and what is critical in the context of the product, as you either design a simple and, let me call it, inexpensive release method that you can apply, that gives you not necessarily detailed characterization, but if you have a stable process, you're not worried about it because you've done that before. So you also have to take into consideration, what for are you measuring it? Are you doing the batch comparison, where you have a standardized system in place, where you know what your risk is and what your variability is because you've characterized it before extensively? That is fine. You just need to provide the science to justify that. At the end, many of the innovations are driven by pragmatism, and we also have to be resource conscious. So the agency is not going to make you do unreasonable things for no good reason. So I think that needs to be considered, and I also believe that with anything that the agency does, more and more so, people understand regulation is also a learning system. Regulation is there to provide certain standards, but also to encourage and give access to the best knowledge that's available in a particular area. And often regulatory agencies have to play that role because they are responsible ultimately as a societal function for safety and quality of product when it goes into people. So, but it's a dialogue, and if new products are being developed and new technologies are being developed, it's not to say that it's going to be static and it's going to be 3 static methods that you can always apply and you're always right.
Any other comments on this topic? Oh, go ahead.
David Schmidt, CSCM, Switzerland. I'm coming from a different point from physical-chemical analysis of nanomaterials. We're working in a European H2020 project called ACE Nano, and we're trying to assess nanoparticle risk with physical-chemical methods. I'm just interested if these new methods could be interesting for clinical nanomaterials. We look at the hydrophobicity of nanomaterials with hydrophobic interaction chromatography, then the solubility with single particle ICP-MS, where we look at the charges, and then the reactivity with a catalytic reaction assay, where we look at how nanoparticles change the reactivity of biological media. And so far we have relatively good results, but it's an ongoing project. And we just look at how the biological media behaves and how it changes as soon as we add nanoparticles. Could this be interesting for highly complex nanoparticles as you're using them?
Yeah, I can comment. Okay.
I think indeed I'm familiar about ACE Nano project. So, and you also use an A4F as well as one of your technique. So definitely the more knowledge is actually presented, the better it is the development of a nano product for medical application. I presume your basis is more a manufacturing material as per se, but the lesson learned is valuable, and the techniques and the development of standard operating procedure are actually very important. Because if these are not consolidated with calibrator, with reference material, then there is no transfer over to different application. Obviously, in our case, we are dealing with a complex system where you have an active component. And the way that is packaged determines how you have to measure it to not compromise it or to actually expose it and then measure it. And as you saw from the presentation, we have also different needs in terms of assessing, the molecular chain and the density of the packing versus the complexity of the hydrophobicity versus the protein corona formation. So it is valuable, and it could be a dialogue that could be open from that perspective. Again, with a view of doing risk as well, because one of the aspects that also Frank has highlighted is safety.
Quick comment. Well, I know it's Nano and your CSCM. And one of the properties that you mentioned, definitely it's interesting. It's the hydrophobicity, and this is something where still we don't have a good answer how to measure the hydrophobicity of nanomaterials. Another point is that because you're working on, let's say, more industrial material, and because I guess you're working in the frame of the European regulation, there, in your case, specifically, you have to take into consideration what is the definition of what is a nanomaterial according to the European Commission, where it doesn't apply directly in the case of nanomedicine. So, as you know, there it's very, very tough, and you have to be able to measure the number size distribution of your particles in the 1 to 100 nanometer size range. But definitely it's interesting what you do.
Yeah, I applaud efforts where they look at, parameters beyond sizing. That's a good thing, different aspects, because that's what we all try to do, obviously. And as Adriel said, you want to make sure there's standards, obviously, when you run it, which I'm sure you do. You have to run references, things like that. But I'd be interested to see, how those techniques can be applied to nanomedicine. So, like Luigi had mentioned, hydrophobicity is something that we're still trying to come up with methods for. I don't know any methods for that per se, so.
So just because you mentioned one thing and then, okay, I think the word standards and the reference materials comes very often here. So I think that would be a good, and definitely us as GRC, but also NIST and other organizations, we are sort of interested in that. Would be interesting for us also to get a consensus or some ideas from the community, because where to start or what are the priorities, especially in terms of reference material, probably.
And David, to further complicate your life, if you haven't grasped it from Andrea's presentation, you need to work in sterile. So you cannot work in a dirty environment with LPS. Because the minute you have LPS, you measure in LPS. So that's where the complexity that Jeff referred to on hydrophobicity, and also on active ingredient, on their activity, the functional activity, you have to preserve it, or you need to work with a chemotherapeutic. How do you handle the toxic element associated with? So these are truly points that we are here to discuss on how this can be brought forward, because there's not, a set of tools that you apply and in a cascade you get the answer, or there's not a ready-made recipe, because each of them has a specific. If you take the same gold nanoparticle that is being presented in this, you already have work for the next 6 years. Okay, just to define the protocols. Okay, so I think it's important, and it's the point of discussing, developing, implementing, improving it, creating methods that can be, and learning step by step, by starting by simple models that you can use as a reference. That could be the NIST calibrators, and from there then you build in, pegylated, what chain of peg, so you're going to use 5K, use a 20K, depends on the application. Let's not go into the aspect of nanobiomaterials for the different application, if it's a medicinal product versus a medical device, because even in that case you have different definition and you actually comply with different standards for assessment. So it's open, and this is where we want to hear more from everybody in terms of methodologies, characterization, what are the critical important steps, what we did wrong as well, or what we are not doing that we should do.
Do you think people understand the limitations of reference materials, where you can get a, say, polystyrene particle that's 150 nanometers and put that into a DLS, which is light-based, but the property, the light refractive index of the particles are totally different. So, I mean, it's not, people I don't think get that. They can buy a reference material with different refractive index and it's essentially useless.
On this one, I'll defer the question to Fanny because she's expert on this, on physical chemical characterization.
Well, honestly, I think we should put some more effort in showing that to people. So we know that, but people out there sometimes they just don't know how to use some instrumentation, and especially when things seems very simple, the risk is very high because especially with DLS, but that may apply to many other different techniques. If you don't know which is the mathematical analysis behind the calculation of the particle size distribution, how you derive different kind of particle size distribution, and so which are the parameters that you have, the inputs that the system needs to properly calculate that, then you get, of course, wrong results. And yeah, honestly, I think that with the risk of using instruments that are easy to use without proper training is there. The risk also of tracing. We talked before about the tracing the data, also the condition that were used to measure. I mean, the risk of we need to check if the measurements are done properly or not. This is why we always support the use of standard operating procedures, and where, of course, these kind of elements are explained. But not everybody, unfortunately, are using them or know really which are the important elements that you need to do the measurement properly. So that's for sure something that we still encounter. And plus, even with reference material, you have the problem. This is one thing and just is related to the value, so that you have to know the properties of your nanoparticles, which sometimes are not easily measured. So, and people underestimate that. We also have another problem, which is actually to use reference materials that are somehow compatible with the particles that you are measuring, because for sure you can select the size range accordingly to what is the size range of the particles that you are expecting. For example, if you have a polydispersed system but you expect to have a different population in some size range that you know, then you may select the polystyrene particles or another standard in the same size range, but they may not have the same chemical properties of the material that you are measuring, and with some technique that can be critical. So this standard can be or cannot be applied, and so that also depends on how this is similar to your system, and unfortunately we don't have standards that are similar to all the complex systems that we are measuring. This is another very important point. But of course, you have to start somewhere. So, for example, we always suggest, if you want to know if your technique has the resolution that you need, to start somewhere. To start to test, for example, a mixture of polystyrene standards that may be similar to the particle size distribution that you are expecting in your system, and at least to test if your system is capable of resolving that. But having said that, that doesn't mean that is the perfect standard for you. But it may not exist. So, yeah.
So, concerning this reference material point, I think we have to really distinguish between a system suitability test, where we use this latex beads, these NIST standards, to see if the system itself allows a measurement in the size range where we at later point expect the particles to be. So we do on a routine basis use 60 nanometer, 100, 200 nanometer beads to see if the system works properly, if it's not dust in which influences your measurement. And then you simply have to adjust your measurement parameters to the system you want to measure, meaning using a different refractive index, using different viscosity values, and so on. And at some point, when it comes to clinical studies, you have to validate your measurement, meaning that you take your samples, add impurities, identify the optimal dilution range where you get stable results. And this can just be done on the final drug product and not on the in-process samples. I think this is something which is completely different. And this is the pragmatic approach from the industry.
I found your comment really interesting. And then I can tell a couple of points where EUNCL already can help and hopefully will help more in the future. So what Fanny was referring, we have published our SOPs for how to measure DLS, and they are public on our website. And over there, sorry? And the NTA, sorry, yes, I forgot about that. And over there, you will see not just the SOP, that's obvious, but you will see we report all the critical parameters that are needed for the analysis of the data. And so that's a very good starting point to see, you need to input, you have a different material, you need to input a different refractive index or absorbance coefficient. On what you said, actually, I hope in the future, UNCL will provide this type, the training, especially if there is a request from the community. I never thought too much about that, but actually, your point is really good. And we have the expertise, and we have the technology, and we have their materials. And so if there is a request and the need, we can discuss about that.
I think the issue is, for me personally, not that the EUNCL can help us, but the other way around. I'm not being immodest about that, because the solutions to those things have been around for 5, 6, 7, 8 years. They're just sitting in a corner in Jeff's lab and other. They're actually quite simple. They're not even complicated. So measuring liposomes and measuring liposome number and measuring the accurate distribution of liposome has been available for at least 10 years. It wasn't a new technique invented last week. I mean, it's really, really quite simple. And I get a little frustrated with the leadership where they perpetuate the use of DLS at 78%. And it's like people are too frightened to say, people, just stop using it. It's just useless. And you people, the leaders in the field, ought to be standing up and saying it, but it's like you're too frightened to upset everybody, to upset the manufacturers or whatever it is. But there's so many complexities. I mean, you start using the FFF system, you see a tower of equipment there, and how long does it even take to do that? I mean, that's not convenient and quick anymore. So I really think you need to rethink your strategy about what techniques you're using, because there are simple techniques out there that solve those problems quite simply. So I'm speaking at 2:15. You should come and listen, I think.
No, it is very true, and as he said, from the manufacturing perspective, you can actually spike your sample and do simple measurement as well. In some cases, we work with sample that we just needed to be spiked, and you look at the relative difference. You can look at the thermal drift. So there's a lot of techniques that have to do with simple measurement. But what we are working with within the framework here is the fact that there's still a mandatory requirement for sizing, and DLS, FDA and EMA speaks DLS first. Show us the DLS.
Correct.
Right or wrong.
That's the problem, but that's exactly the problem.
But I agree, but this is why we're educating. This is why we have a Frank in the session here discussing and providing information. Our slides are going to be sent to them. So the education process is on the mutual discussion on the science evidence. And unless we provide good science where we show the benefit in specific cases, they will go back to what is easy and what they've been training for, because they don't want to spend the time to understand what is a tower of instrumentation can actually deliver to you. So this is what we are doing, and this is not the technique, is the educational system that we are bringing in technical terms with proper understanding and key references. So I hope this clarifies the aspect that it's not that we are perpetuating the same technique, it's just that we are adding additional techniques that could go on top of what is the already accepted or what is the everybody has in the lab. Everybody has a DLS in the world, almost. And this is why Malvern is so rich.
How do you get past that problem? It's like a circular argument. So everybody has it, let's keep doing it, as opposed to the FDA could say, I think they are actually, they've got to grips with it. They're now going to specify a minimum level of resolution for size, which clearly DLS will never meet.
I think,
Yeah.
Okay,
if
so
I
we
may
should try to run out
wrap
of
it up,
time, actually.
I think
so-
we can bring it after because
Yeah.
Vasco Flex has something very interesting to show us this afternoon. So can I take you on this point and saying we have an open debate that is hot now, and we gone to the plenary, we got a coffee, and then from 2 o'clock we continue from this perspective for the one that want to continue discussing into this. We can go on for weeks if you want.
Yeah, so we're out of time, but I want to thank everyone for attending. We can continue this discussion actually in the afternoon session, which focuses actually on instrumentation and measuring particle sizing. So please come back for that this afternoon. Thank you, everyone.